Good afternoon, everyone. I'm going to try to get started here. I know that people are coming back from break or swimming or whatever good things they're doing. My name is Colleen Delaney, and I'm the chair of this next session. We only have two speakers this afternoon. Unfortunately, our third speaker had a family emergency and couldn't be here. So I think both Iago and I just added a couple slides. I'll try to give a little bit of an introduction, but we're going to be talking about cord blood-derived immune effector cells. Okay. So if you can bring up the, do I need to do this? Okay, is this one's my slide? Yes, okay, excellent. Here's my disclosures. I am employed and have equity and am an inventor on some patents held by Derived Therapeutics. Okay, so I'm going to give a little bit, I'm going to take some liberty and talk a little bit about, just give a little bit of background on allogeneic cell therapies and some considerations. I think the sessions yesterday that you heard, especially the one on expansion technologies, and then this morning with Peter Marks was fantastic and a good setup for this. But then also I'll give a couple slides and then I'll just go into my talk and then we'll follow that by Dr. Nieto's talk. Okay, so this is a slide from a recent paper showing the growth of cancer cell therapies. And you can see that there has been steady growth over time. And again, this is just in the area of oncology. And I just want to point out a couple of things, right? We can see the CAR T cells, which we all know, right? There's tremendous growth in this area. But you can see all the variety of different immune effector cell type cell therapies that are being developed, ranging from CAR Ts, NK T cell therapies, different novel T cell technologies, other TCR type technologies, TILs. So it's a very exciting area to be in. I'm gonna talk a little bit about generating monocytes and macrophages off of a platform. So that would fall under this other cell therapy category. But there's been very robust growth, and you can see really in the active pipeline, over 2,000 in 2021 versus 1,500, so a 38% increase in this pipeline. Of course, most of these therapies are still in the preclinical and phase 1 stages of development, and you can see you can barely see, there's a little greenish yellow line here for marketed therapies, and phase 3 is still very lacking. But this is very exciting for all of us in this landscape. Also from this paper, looking at allo versus auto by stage of development. And again, auto cell therapy still very much outnumber allogeneic cell therapy products, but I think many of us would agree, or most of us would agree, that allogeneic cell therapies are very attractive for a number of reasons, and we can see when we look just at the allogeneic cell therapies that we are seeing an increase in the preclinical and phase 1 developmental stage, as well as phase 2. So we're in a pretty exciting time now, and I'm hoping that a lot of the things you've heard here are going to get you excited about allogeneic cell therapies. The advantages are numerous. The use of allogeneic cell therapies overcome many of the limitations we've seen in auto cell therapies. Of course, CAR Ts have really changed the landscape of how we think about treating patients with cancer. This has really been an amazing proof of concept that we can cure, well, actually not a definitive concept that we can cure patients with cancer, but there's been a lot of limitations to this approach that allogeneic cell therapies can overcome. So when you do allo cell therapies, you can really think about the quality of your starting material. There's ability to scale, and then that means having fully released products after you've made them, manufactured them. So we can do on-demand use, which from the patient's perspective, that's incredibly important because it means that they can have timely delivery. There's lower cost, right? Because maybe you can make multiple products per manufacturing run, and this really adds up to improved patient accessibility. We can optimize the quality control for consistency, reliability, efficacy. These are very important for regulatory issues and hurdles. The main disadvantages are always going to be the discussion of the risk of graft versus host disease, alloimmunization, so in other words, are we sensitizing these patients to other donors for future therapies such as stem cell transplant? And then of course, anyone in the allogeneic cell therapy space has to be concerned about poor in vivo persistence, right? Or how quickly are these cells going to be rejected? So what are the considerations when you're developing an allo cell therapy? We've been doing this for a very long time. In fact, we've really only worked, my group has only worked in allo cell therapies. Your starting material, I'm going to talk some more about that on the next few slides. There are a lot of considerations in terms of process development and manufacturing, and the need here is for very consistent generation of, and you'll hear this as a repeated theme throughout, a functional cell, cells that you get at the back end. They need to be potent and well characterized. They need to be scalable because this will help reduce your cost of goods. Cryopreservation is key for this type of cell therapy to be able to distribute easily and, again, for patient accessibility. And you have to maintain your potency and your viability post-thaw. And then, of course, potency itself, right? That assay, and any of the discussions, especially around regulatory, you're going to be hearing more and more about potency assays. And this is key to ultimate approval, and I would argue that it's never too early to start thinking about a potency assay, even though you may not need it in phase 1 of development. But this is going to become more tricky as time goes on. And I think there will be consideration of using maybe more than one potency assay for a particular product. Okay, so what about cord blood as the source material? So not the final drug product, but the source material for cell and gene therapies. So we, in my group, always talk about what a beautiful source cord blood is. And of course, I feel like I'm preaching to the choir here about this one, but we believe that this is a very sustainable, cost-effective, and the youngest source of donor HSC material that's available. As you've heard over and over, there are hundreds of thousands of units that are qualified for use. When we're developing these cell therapies, we can even think about the inventory and what inventory is not going to be used for transplant. Of course, the goal is not to take over the best quality or the highest dose units that are used for transplant, but really to think about how to utilize that inventory. We're doing pooled donor manufacturing, which you may have heard about yesterday when Joe Blake gave his talk, but I'll talk more about that today. And then we believe that cord blood has greater proliferative potential than adult-derived source HSC material. And another very big advantage is that cord blood is now highly regulated, right? It's a drug under the 351s. It's well-defined donor eligibility, and really this is in contrast to adult-derived material. We heard a little bit about this morning in terms of what's a relevant communicable disease, right? So what's considered, what IDMs does one need to do for your starting source material? And I think with cord blood, that's been very well defined. In contrast, though, if you're doing adult sources of cells for your cell therapies, this still seems very variable. I've talked to many people who the FDA may say, well, you need to find a donor that is HHV whatever negative or EBV negative, and that's pretty hard to find. So, and this was brought up this morning as well. So I think, again, cord blood has a huge advantage there as well. And then, of course, the storage of cord blood units is also validated because of all of this work. And I just put a thank you to Hal there because we heard yesterday morning, it's all running into one to me, that now they've looked at 27 years and have shown that cord blood units can be stored for that long. Okay, so that's my little preamble about allogeneic cell therapies and how exciting a space this is. And now I'm going to talk to you about the development of multiple different immune effector cell products from a single platform. Okay, so many of you may have heard me speak before. I'm going to be talking about a platform that is derived really on what's shown here, the Notch signaling pathway. I've talked about this numerous times in terms of the development of expanded HSCs, and today it's a little bit weird for me because I'm going to be focusing more on our immune effector cell platform, but this work was really, the seminal work was done by Dr. Bernstein at the Fred Hutch Cancer Center, and this work stems back to about 30 years ago. So he was my mentor when I was just a wee fellow, about 20 plus years ago, and he had recently discovered that Notch signaling was an incredibly potent predictor or regulator of stem cell fate, right? So we know this is supposed to be showing you the bone marrow microenvironment where you have hematopoietic stem cells. There are cells that line the bone marrow microenvironment that express these Notch ligands, and he made this discovery that the Notch receptors on the hematopoietic stem cell, when they are in contact in the niche with these Notch ligands, that really promotes self-renewal versus different, so it inhibits differentiation, and then you can see that Notch signaling as you get farther out becomes decreased, and that is a very important piece of this. So really when I joined his lab, they had taken that finding in mice and really shifted towards the use of exogenous Notch ligand. So really thinking about how do we recapitulate the bone marrow in in the lab, effectively. And so they developed some proprietary engineered notch ligands, which we still use today. There were many of them that were initially designed. We use one that's called delta X to IgG. It's the extracellular domain of delta 1 fused to the FC domain of IgG. And so this is literally adherent in a tissue culture dish. And this really tries to replicate the bone marrow microenvironment. And we know that we can use this, we can leverage this receptor ligand pairing to control not only just activation of endogenous notch signaling, right? So there's no genetic modifications that has to happen with these HSCs that when we put them into culture, you just use endogenous activation, but it depends on the density as well. So there's a sweet spot to get these cells to self-renew, but it inhibits their differentiation, so we can get more cells without them differentiating. So that was key. And when I started this work in the early 2000s, really what I was after at that point in time was the development of very high numbers of rapidly repopulating cells that I was optimizing for HSCT, so stem cell transplant, particular cord blood transplant. That is not going to be the topic of today, but I do feel like I need to you heard a lot about this yesterday, but just in summary, this platform has been in use for a long time. Our first patients were infused in 2006, and we went from a patient-specific expanded progenitor cell product in double cord transplant. So our group is really the first to show you could ex vivo expand these stem cells to clinical benefit. The time to engraftment was down to 11 days, and that was because this was a matched product. And we did chimerism starting on day 7, so we knew that initial wave of engraftment was from our expanded product. We also knew that these cells had the capacity to do long-term engraftment, so we knew that at least a portion of our product was a long-term HSC. However, it was very logistically difficult. It meant that there would be poor accessibility to patients. That was very important to me, and it was very expensive. And so, I'm getting louder. So we decided to develop this product as an off-the-shelf and cryopreserved HSPC product. So effectively the same idea here. We have no T cells in there, and we developed a product for use as an adjuvant graft. So we got away from the idea that we were going to use expanded cells for long-term engraftment. These were going to be temporary engrafting cells that would bridge hematopoiesis. We did our initial studies in cord blood transplant, again as an adjuvant graft. Dr. Milano talked about that yesterday. And then we also looked at post-chemotherapy, so the neutropenic period after intensive chemotherapy in AML, and we wanted to show safety of using these cells as bridging hematopoiesis, not transplant. And we were able to do that. We showed safety and preliminary efficacy. So in cord blood transplant, we had reduced time to neutrophil engraftment, reduced graft versus host disease, and improved survival in our small initial study. And then in AML, we had a very nice study with preliminary data looking at reduced infection. So that was very exciting for us. In the meantime, we ended up initiating a study with NHLBI funding. Again, that was talked about yesterday, and this was a randomized controlled phase 2 multi-center study of our off-the-shelf progenitors in cord blood transplant. But as Filippo talked about yesterday, we didn't see any difference in the time to neutrophil recovery, and that was really because the standard of care arm was down to 20 days. And interestingly, in the talks we heard yesterday too, other groups like Gamida and their control arm, the time to neutrophil engraftment was also 20 days. So we're getting a sense that as cord blood transplant has gone on, we're just better at picking donors, and we now have lower time to neutrophil recovery. So we did not meet the endpoint in that study. And in the meantime, we worked on scaling of our manufacturing process. So we went from single donor manufacturing to pooled donor manufacturing. And then in 2018, we did the first protocol using pooled donor derived products, and this was the AML study that again was discussed yesterday. We're very excited about this data because it does show that these patients who receive the product have improved remission rates, and the next trials are going to be focused on using this pooled donor product, not in transplant, but in patients with AML, and then actually at the Hutch with Filippo to enhance GVL and cord blood transplant, and he talked about an HIV patient coming as well. So that is the first product that was developed, and I wanted to spend time on that because it's a very important background of safety and clinical data as I talk about the products we're now developing. So this is an overview, a high-level overview of our platform, which is now really truly designed as a platform to derive multiple cell types. So again, we start with proprietary ligands. We always use cord blood CD34, and then I've already talked about dillanubicil, but what I want to talk about today mostly is the natural killer cells and the monocytes and macrophages we can now generate off of this same platform. So we've worked very hard on trying to standardize our approach, right? This is very important as we think about moving forward in our manufacturing process and how to get these therapies through all of the R&D that we have to do and get them into patients. So on the left here is our standardized universal expansion approach. We always use pooled donor manufacturing, so yes, we do have exemption from the GTPs. So we have 3 INDs now around using pooled donor starting material. We do not look at HLA, we do not look at ABO, we only really look at the CD34 content in that product. And then we isolate, as we showed you before, we isolate the CD34s, and then we expand them. And the beauty of this side of the equation is that there is a lot of flexibility built into this platform. So we can do this expansion, standardized expansion process, and we can stop there, that's dillanubicil. Or we can say, well, we want to then differentiate this heterogeneous mixture of cells into an immune effector cell. And we can do NK cells, and we can also do monocyte macrophages, which I'll show you today. And we then end up changing the process a little bit to prime towards the end of the expansion phase in preparation for lymphoid development. So the other side of the equation is a very standardized post-manufacturing process. So again, you have to be able to cryopreserve. We're very good at doing this, and we've shown that we can maintain potency and viability in all of the products that we make. And importantly, and we've done this now in the studies that were talked about already, these are you can hold these products on site for on-demand delivery, and they are literally 20 cc aliquots of cells that can be infused over minutes at the bedside. So what does this lead to? A simple supply chain, very scalable process, lower cost of goods, increased yield, and we think reduced regulatory risk. Okay, so the unmodified NK cell program is where I'm going to spend most of the time, and I'm going to go through how we develop these NK cells and show you some of the data, so hopefully this isn't too data heavy for you all. But as you guys are all very well aware, NK cells are very attractive as an immune effector cell therapy. This has really been shown in many, many years of work well before me, especially in the setting of AML by Jeff Miller and others. And we can see that NK cells have a number of direct and indirect mechanisms of target killing. They are attractive because they are our first line of defense, not just against cancer or cancer-transformed cells, but virally infected cells. They can lyse without having to be educated previously. They secrete cytokines. They can be used without HLA matching as an allogeneic cell therapy. There's a proven safety record with, I think there's a rare report of CRS, but really no GVHD to date has been shown. They can be engineered, which is also quite attractive, and we can cryopreserve them. So these are all the reasons that we are going after NK cells as an immune effector cell therapy. This one is here again to kind of pull you back in and remind you that we have a lot of experience in our expansion phase. So this is really our core technology where we can expand these cells. We've done extensive work on the quality and non-clinical side of things in the generation of the expansion platform. So there was a very nice paper published a number of years ago now showing epigenetic fidelity. So when you take a stem cell, put it in this platform, it is very faithful to the expanded cell you get off of that platform. That's very important from a safety measure, although again, as an allogeneic product, these cells do not live long term. And then, since we've done all these clinical trials with this product, we have a nice foundation of safety with this and lots of characterizations, and we've talked to numerous agencies about this platform. Okay, so how did we optimize for NK cell generation? So again, stem cell progenitor cell expansion and priming in this case. So we've optimized media formulation, we've optimized cytokines during this expansion phase and differentiation phase. We are animal component free. The other thing that's not right here is that we are feeder cell free. And then we also optimize the timing of each of these phases, especially in the NK cell differentiation. These are just figures below showing you that we've looked at many different formulations of media and have shown differences in fold cell expansion. And then we also looked at cytotoxicity, right? So how well do these cells kill? And that's really where we generated our quote unquote optimized expansion process. Joe showed this slide yesterday. We do think that notch signaling, so we did try. We said, well, let's just start from CD34, cord blood-derived CD34 cells and just go directly to NK cells. As it turns out that notch signaling during that expansion phase in the generation of these progenitors is incredibly helpful when we then go on to differentiate these cells. So we do use our notch expansion system in the upfront expansion process. This also gives rise to lots more progenitor cells to make lots more CD56 positive cells in the end. The final cell characterization is shown here, and this is important because I mentioned that we do not use feeder cells. So our final product is a little bit different than most of the other NK cell final cell products that we at least know about. Ours is really a mixture primarily of CD56 positive cells, but there is a portion of cells that include primarily myeloid type progenitors, macrophages, dendritic cells, APCs. This is on average, you can see here that about 75% of our cells are CD56 positive. It probably ranges anywhere from like 60 to 85% by the time we harvest these cells. But importantly, during this stem cell expansion phase, and right here when we go to harvest, or when we transition from expansion to differentiation, it really is the combination of the APCs and the myeloid progeny that we generate during expansion that serve as our feeder cells. And that's why we don't require feeder cells. So we inherently make a feeder cell layer during this expansion phase. So that's worked out quite nicely for us, and from a regulatory IP perspective is also quite nice. So what do our cells look like? When the final generated CD56 cells, you can see here, when we're looking at a heat map, look very similar to our adult counterpart CD56 positive cells. The biggest difference is that our cells do not really express a lot of the inhibitory cures, but other than that, they mark pretty similarly. They also express all of the canonical NK proteins that you would expect to see, aside from the ones that I already mentioned, which with the inhibitory cures not being present. So you can see these are all identified down here. Our percent CD16 is a little bit low, but what we went on to show is that when we put these cells into mice with support of human IL-15, so pre-infusion, you can see the CD16 percentage was about close to 50%, but after infusion, these cells really go on to start to express higher levels of CD16, so that was also encouraging for us. And then function, right? So function is obviously very important. We were very interested. We've always been involved in the area of oncology, especially in the liquid tumors, hematopoietic malignancies, and there's a lot of data to support the use of unmodified NK cells in the setting of AML. And so this data is really showing you the functional assays that we've done on our cord blood-derived NK cell product. And here you can see, so these are cytotoxicity assays. For those of you who are not familiar, this is percent cell death on this axis, and the EDT is the effector to T cell ratio. And so what we're showing here against a number of AML target cells is that our NK cell population does a very nice job in killing AML target cells, except for the KG1A, which is a very resistant AML cell line, not just by our NK cells, but in general. And then the other important thing here is this is a 10-day serial killing assay. So what this is really addressing is how well do our cells repeatedly kill. So when we set up this assay on day 0, the NK cells are put, so the effector cells, or NK cells, are put into culture with our target AML cells. And you then, 24 hours later, can look at the percent killing. And you can see a nice killing curve. They all kind of overlap here. And then we re-challenge the same NK cells by adding additional targets. So this is done on day 3, 6, and 9 as well. And every time we do that, we effectively see good killing by our NK cells. So that's sort of the direct killing. And then do they secrete cytokines? Well, the answer is yes. So you can see 3 different cell lines here. So this is looking at secretion of interferon gamma and TNF alpha. This is the cell line alone. So this is a K562, Kasumi1s, and A549s. And in each of these cases, you can see that the tumor cells alone do not secrete these cytokines. You can see a little bit secreted by, DVX201 is the name of our NK cell product. And then you can see in the co-culture, we see extensive expansion or secretion of cytokines in all cases. And so what about retention of viability and function following cryopreservation and thaw? So again, same process here, expansion, differentiation, then we freeze in LN2, and then we can thaw and do an assay. So this figure here is showing you after, here's pre-freeze viability. So this is viability and cell recovery, pre-freeze cell viability, and then post-thaw viability and recovery. And you can see that viability is quite good, as is recovery for our product. And this is obviously very important if you want to be able to distribute these products for clinical use. Looking at the cytotoxicity assay of our cells against Kasumi-1 target cell lines, again, pre-freeze is this figure here, and you can see that post-thaw, there's a little bit of a lag, but they do catch up, and this is the E/T ratio on the bottom. So we can see good cytotoxicity, and again, a 3-day serial killing assay, so with repeated exposure to our target cells, the NK cells can still effectively kill. So now we've characterized these cells, they are potent, they retain viability and potency after cryopreservation and thaw. So what about in vivo? So we developed 3 mouse models that are frequently used for AML. So this is a Kasumi-1 mouse model, a THP-1 AML mouse model, and then a U937. And in each of these cases, what we're showing you is the in vivo data. This is looking at the flux over time, so looking at the tumor growth by Luminex, and you can see here that so the mice all receive either buffer, so the tumor cells alone, or tumor cells plus our product, and in each of these cases, the infusion of our product delayed the death of these mice, so controlled tumor longer than without the NK cells, and you can see here are the survival curves here on the bottom. Okay, so this is the one slide I have on the use of NK cells as an antiviral. So this was something that we weren't necessarily going to be embarking on. However, when the pandemic hit and we were right in the middle of all of our manufacturing and learning how to make these cells and asking how we were going to use them, there was an opportunity to look at this, and we felt that this was not just an opportunity, but perhaps an obligation to really think about use of NK cells as antivirals, not just for COVID-19, but this presented itself as an opportunity. So why would we do this? As we talked about before, NK cells are really our body's first line of defense as an antiviral mechanism. There were a lot of papers that came out in the early phases of the pandemic that showed NK cell numbers were not only low in a lot of the patients that progressed with COVID-19, but that this correlated with disease severity. And the thought really was that, and the NK cells that were circulating were very dysregulated and dysfunctional. So the thought was, kind of like convalescent plasma, you have a patient who gets infected with COVID-19, not only do their NK cell numbers drop, but many of them become lymphopenic, and the idea was to infuse a source of healthy exogenous NK cells to overcome to overcome the viral infection and prevent disease progression. So, and even today, I guess this is a bit of an overstatement. I wouldn't say there's no off-the-shelf therapies. There are obviously a lot of therapies being used, but there are patients who still, despite the monoclonal antibodies and other therapies and vaccines, who will be hospitalized with moderate to severe COVID-19. And what we were finding, we have a study now, we have 2 INDs for each of these indications, and we are finding that the patients who need these the most are the ones who are immunocompromised. So we did move forward. So we, in the last year and a half, I guess, we now have written 2 INDs, had 2 INDs approved through the FDA. The first is for the indication of COVID pneumonia with Dr. Josh Hill at the Fred Hutch Cancer Center. We have now infused 9 patients. So we did an MTD, a single dose MTD 3x3 design, and all 9 patients have been enrolled. And so now we're about to expand to on the highest level and enroll a few more patients at that dose. We've had no DLT, so importantly, this is a patient population that there was concern by giving NK cells, you would actually make cytokine storm worse in these patients. So these patients we have criteria that they cannot have evidence of true cytokine storm before they get the cells, but we've had no CRS, no infusion toxicities, and we're in the middle of doing some clinical correlative studies. And I'll just mention here too that looking at persistence is really quite difficult in this case because this is a product that is derived from about 6 to 8 donors. And so we're working with a local group to develop an assay to be able to track those 6 to 8 donors in these patients. So we're still working on that. And then we have an ongoing trial in relapsed refractory AML and MDS. Dave Rosieri was the doc who originally helped me with this. He's now moved to Novant, but we are working with him still as the study chair, and Dr. Tom LeBlanc at Duke University, and we're about to infuse the third patient on that. So we finished the first dose cohort, and we're moving on to the second one. Okay, so just as a summary, right, I've shown you that we've been able to generate NK cells from cord blood-derived CD34 positive cells using our proprietary Notch-mediated cell expansion and differentiation platform. We have 2 INDs now. We retain potency and quality, and we're in the middle of these phase 1 trials. Okay, so modified NK cell program. I'm not going to talk a lot about this because I encourage you to stick around at 5:15. Devika from Devera will be talking about her abstract, which is exactly on this part of the platform. But I do just want to say here that with this, again, I talked about this already, but this platform really lends itself quite nicely to a lot of optionality on when and how we will engineer these cells. She will talk about the fact that we do have in vitro and in vivo proof of concept data that we can really do this in the early phase. It does not impact the number of cells we generate, so their growth is the same, their function is the same, despite the modifications that we do here, and they still express the target at the end. But we're also developing and working in collaboration with a number of groups to think about non-viral mediated ways of transducing cells. So please come to Devika's talk. She'll talk about all of that stuff. Okay, so in the last few minutes, I just want to address our myeloid cell program. I think, like most folks working in immune effector cell therapies, we do believe that engineering these cells are going to be a way to increase potency and efficacy for these products. So not only in the NK cells, we're also going to be working on engineering our myeloid cells, but we have just really completed optimization of the generation of these cells, and that's what I'm going to talk about today. We don't have any data yet on the engineering. But again, I feel like I sound like a broken record here. We have a nice platform. So just like in the NK cell platform, we start from this expansion process. Again, we can engineer here, we can engineer later. And this really has, we're differentiating macrophages from pooled donor CD34 positive cells. The advantage of this will be very evident very soon, and really is because when you take monocytes, if you start from monocytes don't really expand in culture, but what we're doing is really leveraging the expansion part of this system to generate, a huge number of CD34s that we can then turn into monocytes and macrophages. So that's the big advantage here. This is a slide showing you, again, you can see this common theme: stem cell expansion, monocyte differentiation and expansion, and then macrophage differentiation. This is showing you the phenotype, just a little bit of the phenotyping from day 0 all the way through. Obviously, on day 0, you expect to see a more purified population of CD34. This is shown in figure here, where CD34 is very high and obviously comes down to near 0, if not 0. And then the HLA-DR11B positive cells will go up, and that's what these dot plots are showing you here. So we've, again, optimized media formulation. That's been quite interesting. We've optimized cytokine combinations, again, animal component free, and we've optimized duration of this. This is just showing you some additional markers that we have looked at as we've been making these cells. And interestingly, depending on the formulation, we can really skew towards pro-inflammatory, as shown here, or anti-inflammatory. So depending on the process that we use. So these cells can be cryopreserved. We were quite excited to see how well we were able to do this, and we can thaw them with recovery of viable cells. This here is showing you fold expansion. So at the end of the expansion phase, you can see generally we have about 250, 300-fold expansion, if not more, of the CD34. And then at the end of the monocyte phase, and this data is a little bit older now, you can see that we have a nice expansion of the monocytes, and then from monocyte to macrophage, there's very little growth, so most of it happens here. And you can see that most of the cells do mark as monocytes and macrophages, and then post-thaw, we retain recovery and viability. Again, there's updated data. There's a poster tonight on this data as well. I'd encourage you to come see that. We've now been able to get our fold expansion upwards to close to 100,000 fold expansion. So come take a look. Do they function? The answer is yes, they do function. This is looking at incucyte data. I've got a cool movie here, right? So when these cells phagocytose, they become red, but this is just the graph of this looking at Kasumi cells that don't phagocytose, so in black, you don't see any phagocytosis. This is a THP1 cell line that is capable of phagocytosis, then our monocytes, and then our macrophages. And let me just show you the movie. You can see that the cells, by the end, will be mostly red. So we have nice functional data for that as well. So the overview really is that, again, we can generate functional cells. We can generate a lot of them. We have worked on media formulations that can influence how we polarize and what kind of cell we generate. We're continuing this optimization work, and we're really looking at this now with an eye towards working on engineering these cells in the future. And so come see the poster. So finally, hopefully I've shown you that really what we think is pretty cool about this platform in particular is that we've standardized an initial stem cell expansion process prior to differentiation. It's very unique because this whole process has been used clinically, right? So we have a clinically proven pooled donor stem cell expansion process, right? So we have put pooled donor products into patients. It's a safe starting material without genetic alteration. We can do very consistent, Joe showed this data yesterday in his talk about the significant reduction in variability from lot to lot by using pooled donors. We generate highly functional, potent, and well-characterized immune effector cells. They retain viability and potency. And our ultimate goal is really to think about personalized approaches using these immune effector cells so that we can really try to mimic the natural immune system by using multiple different products at the same time, especially with engineering. Okay, so with that, hello. I want to just thank, so everyone at Derived, we have a small team. I think Monica, I don't know if she's here, but she said yesterday that you have a small and mighty team. I say that a lot. So I heard you loud and clear when you said that. So thanks to everyone at Derived. And the Hutch is obviously where all this work started. And we really couldn't have done a lot of this work without the early work and even our ongoing collaborations, including the mouse facility and the GMP facility where our NK cells are currently being made. I'd like to thank the clinical teams, so Dr. Josh Hill and Dr. Rizieri and Dr. LeBlanc. Oh, I forgot his MD. Sorry, Tom. The clinical research teams and the patients who are enrolling on our studies. And finally, we're working very closely with Cleveland Cord Blood Center, not only for sourcing cell material, but hopefully for future manufacturing as well. So please come see the posters. And with that, I think what I'll do, actually, I can take a few questions. I'm trying to figure out I'll take a few questions, because since we only have the two speakers, if anyone has any burning questions, I can do them now, or we can wait till after Dr. Nieto goes. Yeah, Gesine. I can hear you, but they can't. How long does it take to generate the monocytes? Well, so that's an interesting question because initially it was about a 2-week expansion, which is our normal, and then about a week and a week. But we've actually been playing with the expansion phase because the more cells we can generate there, the more monocytes we can generate. So it's, Devika, 4 weeks, right? Give or take. For the NK cells, it's about 24 days. Okay, I'm gonna let you guys all stew. Oh, wait, okay.
So I was wondering what clinical applications were you contemplating regarding the monocytes and macrophages?
So the question was, what clinical indications are we contemplating with the monocytes and macrophages? That's a terrific question. I don't think we really know at this point. I think it's more clear with the NK cells. We hope to be going after hematologic malignancies in some, in the modified NK cells. With the monocytes and macrophages, we are very unlikely to go after an autoimmune type process. We really think that we can engineer these cells to be used in concert with potentially engineered NK cells. So the idea, hopefully with solid tumors. So can we do a kind of mark and kill type of strategy where we can engineer our myeloid cells to attract the immune system, either of the host or of some other modified cells? But it's a little bit unclear still. It's a great question though. If you have any ideas, I'd love to hear.
I see, thank you. One more question, if I may. Have you assessed the capacity of these NK cells to perform ADCC using maybe current marketable antibodies?
Yeah, so a little bit. Not a ton of that work has been done. And we also talked about, like many are doing the, engineering the cells to have the high affinity CD16. So we haven't done a lot of that work yet. And we're really thinking about going after some other indications. But yeah, another good point.
The best of luck.
Thank you.
Hi. Really nice work.
Thanks.
I was just wondering, so the NK cells, they don't express inhibitory receptors. So do you see licensing happening after transfer and no licensing happening in the dish?
So that's what we want to figure out, right? And that's going to be one of the things with this assay that we're trying to develop. Can we pull out some of the cells that have been infused? So we've been collecting samples from all the patients who have received these cells to try to ask some of those questions. We also tried to look, because one of the thoughts is where they're just not they're fully potent, but they don't seem to be fully mature. And so when you put them in the human environment, are we going to start to see that maturation? So we have samples to look at that, and we're just starting to do it. But in the COVID population, I'm not, I don't know, but we're going to try to look at that in both the COVID and the AML study that we're doing, and we're hoping that that will help us with future, modifications that we want to make with the cells. Thank you. Yeah, sure.
On the inhibitory ligand question, there is a big question about the potency or efficacy of NK cells with the balance of activating inhibitory ligands, and I think it's kind of still out there to prove. Have you done any comparisons against donors with different ratios of both?
Well, no, we haven't. And the other thing we haven't done a lot of comparisons with is just adult-derived NK cells. So we've been using cord blood, and we haven't screened them for any of that. I think we're also using pooled donor, so that's another question, right? How does that impact some of this? And we're very curious to understand how our cells will behave in vivo. Will some of, so if we use 8 donors, for example, do 2 of the donors persist longer? And what are the characteristics of those 2 donors versus the other ones?
Thanks.
Yep. Okay, well, we have lots of time. So I'm going to introduce Dr. Nieto to come up. And thank you very much for agreeing to speak. I'm super excited to hear this talk.
Thank you, Dr. Delaney. Good afternoon, everybody. So I'm Iago Nieto from the Department of Stem Cell Transplantation at MD Anderson. These are my disclosures of relevance to the talk. The University of Texas MD Anderson has a license agreement and a research agreement with Affimed. So in this talk, I will first discuss the basis for the cellular immunotherapy approach of our group at MD Anderson, specifically why we're pursuing the use of allogeneic instead of autologous products, why CAR-like instead of CAR cells, and why NK cells instead of T cells. Second, I'll present our preclinical results with AFN13 precomplex cord blood-derived natural killer cells for CD30 positive lymphomas, and I'll discuss our early clinical results. So we're all familiar with the revolution that CAR-T, autologous CAR-T, represented in the last 10 years. In the last 5 years, the FDA has approved various products for a number of B lymphoid malignancies. The responses range from 45 to 90%, depending on the disease. And there's no question that this is very exciting, and it really has constituted a paradigm shift. Having said this, there are a number of limitations that come along with autologous CAR T cells, mainly the length of manufacturing, which can go on for several weeks. As well as the fact that the autologous products are patient-specific, and with that comes the problem with patient access and cost. And for that reason, there's been great interest by many groups searching for alternative immune effectors that could serve as off-the-shelf therapies. So my group, our group at MD Anderson, led by my colleague Katie Rezvani, has been pursuing the use of natural killer cells for allogeneic products, such that the use of cells from one donor could lead to manufacturing of hundreds of products. And of course, that is a huge advantage in terms of patient access, reduced costs, and many, many more patients who could benefit from this treatment. So what are NK cells? So as Dr. Delaney discussed in her presentation, these are cells that are part of the innate immune system and the first line of defense against virally infected cells as well as malignant cells. They share the same killing machinery with cytotoxic T cells, but they have a number of unique features that make them very suitable for allogeneic treatment. They don't cause graft-versus-host disease, and their antigen, the target recognition is not through a receptor like the T cell receptor, which is generated through DNA recombination, but rather they have a broad array of germline-encoded receptors which are activating or inhibiting receptors, and it is the final balance for each NK cell between both receptors which will ultimately determine whether that NK cell will kill or not its target cell. So if I can briefly take you through that, when an NK cell comes into contact with a normal cell, and as you know, all normal nucleated cells present HLA class I. The inhibitory receptors engage with the HLA class I and send a negative signal that overrides any potential activating signal from the activating NK receptor that might bind and activating ligands on the normal cell. So in contrast, when an NK cell engages a virally infected or malignant cell, and as you know, these cells very commonly downregulate the expression of HLA class I, when the NK cell comes into contact with these type of cells, then the inhibitory KIR receptor does not bind its normal ligand, which would be HLA class I, and therefore there's no negative signal, and the activating signal from the activating receptor goes completely unfettered. So we don't know. There's many activating ligands for NK cells which we don't know about, but those that have been described appear to be molecules that are expressed on the surface of the cells that result from DNA damage on the stressed cell. So as a consequence of this differential balance between inhibitory and activating receptors, according to whether the NK cell encounters a normal or a malignant cell. As a consequence of that, the NK cell can kill malignant cell and mediate tumor responses, but does not induce graft-versus-host disease on normal cells. So it is quite clear to us that there are a number of advantages of the NK cells for CAR therapy over the T cells. So they're more suitable for allogeneic therapy. They don't cause graft-versus-host disease as opposed to allogeneic T cells, at least those that have a TCR on their surface. Their killing is mediated not only by the directed CAR, but also by their innate activating receptors, including CD16, the natural cytotoxicity receptors, NKG2C, NKG2D, and some others. So that means that if the target cell loses its target antigen, it can still be subject to cytotoxicity from the NK cell, and thus that could perhaps reduce the risk of relapse. And finally, and for reasons that are not totally understood, as opposed to CAR T cells, CAR NK cells do not cause CRS or ICANS. And that might have to do with reduced production of IL-6. So back in 2020, my colleague Katie Rezvani first reported on the first in-human trial of CAR NK cells using a CD19 CAR in B lymphoid tumors. So what we did in that trial was we transduced cord blood cells from umbilical cord blood NK cells, which had been expanded in the presence of K562 feeders with membrane-bound IL-21, CD48, and together with IL-2. So these NK cells in the dose escalation phase of the study resulted in 7 out of 11 complete responses with no cases of CRS, neurotoxicity, or GvHD. Now, mature NK cells, in the absence of cytokine stimulation, their lifespan, in blood is around 2 to 3 weeks. So to circumvent that, Dr. Rezvani transduced the NK cells with an IL-15 gene, which IL-15 is a very important cytokine for NK cell persistence and proliferation. And now we know that these CAR NK cells persist, albeit at low levels, up to 18 months out. So CAR engineering allows for redirection of antigen specificity, enhanced persistence by incorporating a cytokine gene. But at the end of the day, CAR engineering is still expensive and technically challenged. So our hypotheses were that pre-complexing NK cells with bispecific antibodies prior to infusion would facilitate CAR-like responses by the NK cells. In addition, the persistence of the NK cells can be enhanced by pre-activation with cytokines to induce a memory phenotype. So we chose the tetravalent bispecific natural killer or innate cell engager AFN13, which is, has been developed by Affimed to test this hypothesis. And you can see here that AFN13 binds CD16A on the NK cell, and on the other hand, it binds CD30 on the tumor cell. So a hypothesis again was that pre-complexing allogeneic NK cells with this bispecific innate cell engager AFN13 would increase the cytotoxic capacity of the NK cells. What about memory cells? So we all know that natural killer cells are part of the immune system, the innate immune system, and on the other hand, memory is a feature of the adaptive immune system. Now, it's been known for the last 15 years that after infections with viruses, and also after exposure to cytokines that are normally produced by dendritic cells, such as IL-12, IL-15, and IL-18, this results in a memory-like phenotype of the NK cells where they have increased proliferation, increased persistence, increased release of interferon gamma, and ultimately increased anti-tumor activity. So with this, we set off to test our hypothesis in the lab, and Lucila Kirby, working at Katie Rezvani's lab, showed very nicely that AFN13 loaded or pre-complexed, but not NK cells, but not those NK cells that were not pre-complexed with AFN13, killed Carpus cells. And this is a cell line that is T cells that express CD30. So as you can see here, the AFM-bound NK cells kill many more tumor cells, Carpus, than the unloaded NK cells. And it's also resulted in increased degranulation and expression of interferon gamma and TNF-alpha. Importantly, the AFM13 loaded but then washed NK cells had similar level of cytotoxicity against corpus as AFN13 loaded and unwashed NK cells, meaning that the the AFN13 exerted its effect when bound to the membrane of the NK cell and not as a soluble factor. This effect was specific, so it did increase the cytotoxicity of NK cells on CD30 positive cells, but not CD30 negative cells. And in vivo experiments showed by Lucila showed that precomplexing cord blood derived NK cells with AFN13 prior to infusion facilitated core-like responses in a CD30 positive mouse xenograft. So the persistence of the NK cells was enhanced by preactivation with IL-12, IL-15, and IL-18 to induce a memory phenotype. The NK cells expanded more than 1,000-fold in the presence of K562 feeder cells. And as you can see here on the right side of the slide, the AFN13 complex preactivated and expanded NK cells induced greater cytotoxicity than the same preactivated and expanded NK cells, but not bound to the AFN13 or to AFN13 alone. Importantly, we saw that retention of AFN13 on the surface of the NK cells lasted for up to 7 days, and that the AFN13-bound NK cells successfully homed to sites of disease in the mouse, such as the liver, the spleen, or the bone marrow. In addition, Lucila showed that the preactivated and expanded cord blood NK cells had presented upregulation of genes related to JAK-STAT or interferon gamma signaling. So this very encouraging preclinical results led us to test this hypothesis in patients. So we thought that Hodgkin's lymphoma lent itself very well to testing of this therapeutic model. Hodgkin's lymphoma is characterized by the presence of very few malignant cells, known as Reed-Sternberg cells, within a reactive mixed cellular infiltrate, which is the tumor microenvironment. So the important thing is that the Reed-Sternberg cells only constitute 1% of the whole tumor mass, whereas the remaining 99% is the tumor microenvironment. And this is probably a unique phenomenon in human oncology. So very importantly, there's a marked immunosuppressive crosstalk between cytokines released by the Reed-Sternberg cells and receptor on cells in the tumor microenvironment, such as the tumor microenvironment is really immunosuppressed. As a consequence of that, the patient's own autologous NK cells, their function is impaired in patients with Hodgkin's lymphoma, as you can see here, as opposed to the function of NK cells from healthy donors. With regards to the clinical niche that this treatment could have in the management of Hodgkin's lymphoma, the target antigen CD30 is universally expressed in classical Hodgkin and many T-non-Hodgkin lymphomas, such as angioimmunoblastic T-cell lymphoma. It is effectively targeted by the antibody drug conjugate brentuximab vedotin, which is active in relapsed patients, even patients who have failed an autologous transplant, with a pretty high response rate of 75%, including 34% CRs. Now, only 1/3 of those CRs, which comes down to only 1/9 of all patients treated with brentuximab, in only 1/9 those CRs are durable, meaning that most patients eventually will fail brentuximab. Importantly, at the time the patient failed brentuximab, their tumors still express CD30. The other important class of drugs for Hodgkin's lymphoma is checkpoint inhibitors, which induce a very high response rate between 50% and 80%, even after brentuximab vedotin failures, but a CR rate which is half that of brentuximab, around only 15%. And most importantly, in the event-free survival curves, we see no plateau, strongly suggesting that patients who receive checkpoint inhibitors are ultimately not cured. So there's clearly a very large unmet need for patients with relapsed Hodgkin's after failure of brentuximab and checkpoint inhibitors. So this summarizes the treatment schema. So the NK cells are separated immunomagnetically from a unit of umbilical cord blood. We are fortunate at MD Anderson to have our own cord blood bank, which was established by Dr. E.J. Spall, and we have more than 30,000 units now. So these NK cells, from the day the cord blood unit is received, they're manufactured over 2 weeks, during which the first step is their pre-activation with the 3 cytokines I mentioned before, IL-12, IL-15, and IL-18, which will confer on them a memory-like phenotype. Subsequently, they're expanded with the 562 feeder cells together with IL-2. And at the very end of the 2 weeks, they're incubated with AFM13, that bivalent antibody construct in the lab. During that period, while the cells are being manufactured in the lab, the patient receives lymphodepleting chemotherapy with the usual regimen of fludarabine and cyclophosphamide. And again, Flu-Cy is not intended to treat the Hodgkin lymphoma. In fact, fludarabine is inactive in Hodgkin's, and these pretreated patients will probably not respond to any cytoxin at all. But the role of this is very important, because by treating the patient's own immune effector cells, that reduces the risk of rejection of the NK cells, and furthermore, we decrease the cytokine sink for IL-7 and IL-15 that these patients' immune cells would represent. On day 0, the patients receive the infusion of the AFM13 complexed NK cells, and then they receive on day 7, 14, and 21, 3 intravenous doses of AFM13 at a dose of 200 milligrams. The reason for this is that, like I showed before, we saw that the off rate of AFM13 on the surface of the NK cells is probably about a week, maybe 7 to 8 days. So that's why we give boost doses of the AFM13. On day 28, the patients have a PET scan to evaluate response, and this constitutes 1 cycle. So when we first designed the trial, we contemplated treating patients with 2 cycles. This was a phase 1 dose escalation trial, where the doses of NK cells would go from 1 million per kilo to 1 times 10 to the 8 per kilo in dose level 3. We followed a very simple 3+3 escalation rule with a target accrual of 46 patients. So these are the inclusion criteria. I just want to bring your attention to the age range, which goes up to 75 years. And this trial enrolled patients with relapsed or refractory classical Hodgkin's or other T-cell or B-cell lymphomas that expressed CD30 at a level by immunohistochemistry of equal or greater than 1%. Now, importantly, Hodgkin's lymphoma and anaplastic large cell lymphoma patients, those are diagnoses where brentuximab vedotin has shown to be beneficial. These patients had to be refractory or intolerant to brentuximab vedotin. The rest of the inclusion criteria and exclusion criteria are pretty standard. Basically, they just required adequate end organ function. The primary endpoint of the study is to establish the safety and recommended phase 2 dose of the cord blood NK cells. Secondary endpoints included to assess the overall response, complete response, and partial response rate, to evaluate event-free and overall survival, to quantify the persistence of the infused donor AFM NK cells in the recipient, and to study the activation phenotype of donor NK cells. So we have treated 22 patients, and I'll present response rate on 19 of them. Median age is 40, with the oldest patient 75, 15 males, 7 females. Almost all of them were Hodgkin. There were 2 T non-Hodgkin's lymphoma. They were heavily pretreated with a median 7 previous lines of therapy, and 1 patient had received 14 lines of therapy. They had all received brentuximab vedotin. 21 of the 22 had received anti-PD-1 and failed anti-PD-1. The one patient who did not receive it was a patient with T-cell lymphoma, where anti-PD-1 is usually not used. 2/3 of the patients had failed a previous transplant, autologous in 9 of them, and allogeneic in 5 of them. 2 patients had failed previous CD30 CAR-T. They had suffered a median 6 episodes of relapse or progressive disease, and they all had progressive disease immediately prior to study enrollment, and no bridging therapy was used between enrollment and treatment on study, primarily because the manufacturing time is only 2 weeks. So as far as the characteristics of the infused product, more than 99% of the cells were NK, which had expanded more than 2,700-fold. The content in T cells was only 0.02%. The NK cells were 91% bound to AFM13. They were 96% viable, and more than 97% expressed CD16 on their surface. So CD16, again, is not only the target for the AFM13, but it's also an activating receptor, a very important one in the NK cells, probably the most potent activating receptors that NK cells may have. So cord blood NK cells were selected without consideration for HLA matching, which was no matching at all in 17 cycles, match for 1 antigen in 19 cycles, and we didn't see any association between the degree of HLA matching or mismatching with adverse events, response, or persistence of the NK cells. Pre-medication for the AFN13NK and AFN13 infusions included antihistamines and acetaminophen. Specifically, steroids were avoided. G-CSF was allowed for grade 3 to 4 neutropenia, obviously caused by the lymphodepleting chemotherapy, and cycles 1 and 2 were administered a median 56 days apart, counting from day 0 to day 0. So the treatment was very safe. We didn't see any cases of cytokine release syndrome, neurotoxicity, or graft-versus-host disease. We didn't see any infusion-related reaction of any grade in any of the infusions of AFN13NK. We saw 5 infusion-related reactions, 1 grade 3, 4 grade 2, in 108 infusions of AFN13 alone, for a total prevalence of 4.6%. This is substantially lower than when AFN13 was used in its phase 2 studies as a single agent, where the likelihood, the risk of an infusion-related reaction exceeded 30%. We saw the expected myelotoxicity from lymphodepleting flu-cy with neutropenia, which was 63% grade 4, 21% grade 3, and again, the use of G-CSF was permitted after encountering this degree of neutropenia. Thrombocytopenia was less prevalent, and importantly, we didn't see any cases of neutropenic fever or clinical bleeding. We didn't encounter any dose-limiting toxicity, and therefore, dose level 3 at a dose of 1 times 10 to the 8 per kilo was established as the recommended phase 2 dose. So responses were evaluated by PET using lytic criteria on day 28 of each cycle. We saw 17 out of 17 metabolic responses in 19 patients. This includes PRs and CRs for an overall response rate of 89.5%. With 10 CRs and 7 PRs in the whole study file. Now, among the 13 patients who were treated at the recommended phase 2 dose, there were 8 CRs and 5 PRs. And among these 8 CRs, some of them converted from a PR after cycle 1 to a CR after cycle 2. And you can see here on the right the waterfall plot of the responses, which really includes mostly patients with Hodgkin's lymphoma. This is the Swimmer's plot showing the duration of responses, which is really the evaluation of that is really limited by the fact that in some patients we consolidated our response to treatment with a stem cell transplant. However, there's 5 of them who persist in unmaintained complete response at close to a year now of follow-up. So to illustrate the quality of these responses, I'll show a couple cases. So this is a 53-year-old female with Hodgkin's lymphoma, which had been completely refractory to 5 previous lines of therapy, including ABVD, ICE, brentuximab vedotin, nivolumab, and gemcitabine oxaliplatin. So when we enrolled her, she was very debilitated with pronounced B symptoms at enrollment. She was treated at dose level 1, and like you can see here, the baseline PET showed bulky multi-compartmental lymphadenopathy. She had a splendid partial remission after the first cycle, which converted into a complete response after the second cycle. The other case is a 37-year-old male with highly refractory Hodgkin. He had failed 10 previous lines of therapy, including AVVD, GDP, ICE, an autologous stem cell transplant, Pembro, bendamustine, brentuximab, brentuximab nivo, brentuximab everolimus, and CD30 CAR-T. His Hodgkin just progressed to the CD30 CAR-T, and that was the treatment immediately prior to enrollment. He had marked B symptoms at enrollment. He was treated at dose level 3. So this is the baseline PET. Besides the bulky lymphadenopathy, you can see here in his right neck and both axilla, he had also multiple lesions in his lungs and bones. So he had a very good response after the first cycle with just some minor still slightly uptaking nodes here in his right neck, as well as the right axilla, which converted to a CR after cycle 2. So these are the Kaplan-Meier curves. So for the entire study file, at a median follow-up of 9 months, with a lead follow-up of 19 months, the EFS rate is 52%. Overall survival rate is 81%. For the subset of patients treated at the recommended phase 2 dose, the EFS is 67%, and the OS rate is 93%. So we studied blood levels of the donor NK cells, and as you can see here, they experience a peak in blood right after infusion, but the level persists for anywhere between 3 and 4 weeks. Also, very importantly, the NK cells experience the similar pattern of blood levels after both cycles in each patient. And like I said before, each patient received the same dose of NK cell in each of the 2 cycles. And this is important because it argues against sensitization of the patient against NK cells. Probably because they come from different core blood units, and it is in stark contrast with the published experience with repeated infusions of autologous CAR-T cells, where in second or subsequent cycles, despite the use of higher doses of autologous CAR-T cells, their blood level is gradually lower. In a few patients, we had the opportunity to interrogate the phenotype of the donor NK cells, looking for activation markers, and the recipient and donor NK cells were separated by flow using HLA. We studied by cytoff, mass cytometry, markers of activation up to 40, and you can see here on this heat map that donor NK cells present much higher expression of CD16, AFM13, of course, and other markers of activation like CD69, NKG2C, as well as the cytotoxic granules perforin, as well as TIGIT, which is another marker of activation. So moving forward, the study is still ongoing, but how can we optimize this therapy? Well, as responses deepen from cycle 1 to cycle 2, it stands to reason that it may be beneficial to patients to increase the number of cycles, and that's what we're doing now. We're treating patients with up to 4 cycles. We're also shortening the interval between cycles. Just to remind everybody, these are patients with very heavily pretreated and aggressive lymphomas, and obviously increasing the density of treatment will decrease the risk of relapse between cycles. And ultimately, we are looking at combining these NK cells with agents such as lenalidomide, which are known to enhance NK cell function. So in conclusion, This is the first clinical trial using off-the-shelf cord blood-derived, cytokine-induced, memory-like, ex vivo expanded NK cells, pre-complex with the innate cell engager AFN13 construct to treat patients with CD30 positive, relapsed refractory Hodgkin's and non-Hodgkin's lymphoma. Our preliminary results show an excellent tolerability profile with no instances of CRS, neurotoxicity, or graft-versus-host disease. We saw very encouraging activity in heavily pretreated patients with 100% responses and 62% complete responses among patients treated at the recommended phase 2 dose. The donor NK cells persist in the recipient for up to 3 weeks with an activated phenotype, and we believe this approach warrants further investigation for the treatment of CD30 positive lymphomas. So I want to acknowledge all my collaborators, first and foremost, Dr. Katie Rezvani and her research lab, who did all the preclinical work on pre-complex cord blood NK cells, the GMP lab led by E.J. Spall, the MD Anderson Cord Blood Bank, my colleagues in the stem cell transplant faculty at MD Anderson, all the rest of the stem cell transplant department, PharmDs, our mid-level, the research donor coordinators and research staff, my colleagues in the lymphoma department who are very active in referring patients to us, and of course my partners in AFIMED, and of course, and first and foremost, our patients. Thank you very much.
Okay, does anyone have any questions? I'm gonna start while everyone thinks about what they want to ask. You're giving these cells multiple times. You didn't mention cryopreservation, and I didn't see it in the schema. Are you cryopreserving the product?
Not yet. That's the next step.
Okay, so can you walk through? So you do the expansion, but then you're giving them on different days.
Obviously the cord blood.
Yeah.
So obviously the cord blood is frozen in the back. We identify a unit that's suitable to treat a patient, and we have our own algorithms for that. And that unit is thawed. The NK cells are separated, go through the process of preactivation, expansion, and lastly, incubation with AFN13 over 2 weeks.
Okay.
And then they're infused.
Okay.
We want to really move forward to a truly off-the-shelf product in which all of this will be followed by freezing of the NK cells. Now, NK cells are more susceptible to cryo injury than T cells. So we know there's a fraction, maybe around 30% of NK cells that will be lost in thawing. So we need to adjust for that. But that is where we move into next.
And are you matching the units right now to the patient, the NK cells, or?
HLA matching?
Yeah.
No, not at all.
Okay, so that part is off the shelf. It's just, so you can
Yes,
select any unit from
we don't do
the
any
bank.
HLA matching, and we haven't seen any GVHD.
And it seems like you get 2,000-fold expansion. So you must have more than you need at the time of infusion.
Well, I'm a transplanter, so the more the better. The NK cells are expanded with the feeder cells. The median is 2,700-fold. There's a dose-response effect with the NK cells. We saw better activity as we proceeded with the dose escalation in the phase 1. So the most long-lasting responses we saw them at dose level 3. We didn't go up above dose level 3 because technically it's not feasible.
Right.
It's not feasible. We don't have the machines in the GMP lab to do to manage that volume of cells.
Okay.
But we think that the more NK cells we get, the better.
Okay, Mitch.
Really cool work. Thanks for that. As you know, we've learned from CAR T that persistence is key. And I'm wondering, is there any work or any insight as to what cell type is eliminating the allo NK cells? And if so, is there any ideas yet as to how to promote persistence, whether it be with serotherapy, if it's T-cell driven? These are really heavily pretreated patients, so as this gets more, as results are more encouraging, you're going to get less heavily pretreated, and I'm concerned that then persistence will be even shorter.
That's a great question. So we know that, we anticipated that these mature NK cells would last for 2 or 3 weeks, or 3 weeks, because in the absence of cytokine support, that's as long as they can last in a patient, allogeneic NK cells. The subset of immune cells in the patient that are responsible for their rejection ultimately are the T cells, presumably, because those are the ones that mediate rejection of any allogeneic T cells. So there's 2 ways of handling that. One is with the obviously lymphodepleting chemotherapy helps. You decrease the lymphocyte count in the patient, and you suppress the cytokine sink, as I mentioned before. Alternatively, these NK cells could be transduced with the IL-15 gene, like we do in our CAR-NK studies. But this would mean that we would only give 1 cycle. Jeff Miller and his group in Minnesota tried giving IL-15 in vivo to increase the persistence of the allogeneic NK cells, and he ended up seeing that those NK cells, actually the persistence was less, because paradoxically, they did increase the initial proliferation of the NK cells, but they also stimulated the patient's own CTLs, which ended up rejecting the NK cells sooner. So it's a difficult situation, and the solution we have come up with is increase the number of cycles. We know that these cells act very rapidly. We have seen big masses, peripheral masses, melting away in days, a matter of days. It is you can try to make them persist for longer, but then don't expect to give more cycles, because when you give another cycle, you give lymphodepleting chemo, it's going to get rid of the residual NK cells from the previous cycle, right? So what we're trying out to is to, by the use of frozen products, to make it much simpler and increase the access of more patients to a greater number of cycles.
Guy Claimer, City Cord Blood Bank. Thank you very much. Very interesting talk, comment. If they were long-lived, I would have had a question about what would happen with activated T cells that express CD30. So it's probably a good thing that they're not long-lived. Question is about CD16. There are reports that CD16 is dim and lowly expressed on naive natural killer cells in cord blood. Is the expression of CD16 in your starting material an indicator of proliferative potential or potency efficacy?
So you're right that cord blood NK cells, they don't have a mature phenotype. They don't express a lot of CD16. However, that increases with the pre-activation process we do in the lab by exposing those NK cells to IL-12, 15, and 18. So their CD16 expression significantly increases. Furthermore, incubating them with the CD16 specific AFN13 the expression of CD16 go further up. And we saw that in the CyTOF panel I showed, where the expression of CD16 is very high. And this is important. It's very important, because CD16 is probably the strongest activating receptor that NK cells have. It entirely mediates ADCC. So it's a very important observation.
When this goes to become standard therapy, do you think we're going to have the same issues with exhaustion as we do with CAR T cells?
I wish, because that means that the NK cells linger around long enough to become exhausted. I think the main problem is that these NK cells just disappear, which is what Mitch's question was about. So they linger around for 4 weeks. So in 4 weeks, no immune cell is going to get exhausted. In studies where other NK cell therapies have been combined with checkpoint inhibitors to reduce their exhaustion, the results, I don't think they're that promising. I think at least in NK cells, their exhaustion is not mediated by PD-1, PD-L1.
Thank you.
Thank you for your talk, and congratulations for beautiful results. I was wondering, since you are you are both loading the product, you're both loading the NKs with AFM13, as well as administering AFM13 afterwards for at least 3 doses, and then you say that you wish to continue with additional cycles in the future. So I was wondering if you witnessed anti-AFM13 antibodies emerging in patients receiving high doses of the drug product, actually.
We are measuring that. We don't have the results. However, in the phase 2 studies of AFM13 alone, the oldest one was published in Blood. It was conducted by the German Hodgkin's group. So they did see anti-drug antibodies, ADA, but they're not neutralizing antibody. They have no effect on the pharmacokinetics of AFM13.
I see. Also AFM13 engages other Fc gamma receptors?
I'm sorry?
Does AFM13 engage other Fc gamma receptors like CD32? Is it possible that this construct is not only specific for CD16, but you have off-target effects?
No. It's a product developed with a platform that is called ROC. The Germans came up with this fantastic product with very high specificity for CD13 and CD16A. So CD30, I'm sorry, not CD13, CD30. So CD30, as you know, is expressed by a number of lymphomas. CD16A is expressed, as opposed to CD16B, is not expressed by neutrophils. It's only expressed by natural killer cells and macrophages. We think since we are combining it, incubating it with NK cells, we don't think that the part of anti-tumor effect that could be mediated by the patient's own macrophages is very high. We don't think that. As far as the activity that AFM13 might have alone, we know from the German studies that it's very low. The response rate is 10% at most. And that is because if you give this antibody, it's not an antibody, it's an antibody construct. If you give it alone, it obviously is entirely reliant on the patient's own NK cells, which are dysfunctional to start with, right? So you're expecting to get a good response from the patient's own innate immune system, which really is not, at least in Hodgkin's, is not functional.
I see. Thank you very much.
Just a quick question about the cord blood units that you're using for these therapies. You said HLA wasn't a requirement, so I was just curious about your thoughts on, or your strategy for, selecting the specific cord blood units. Is it based on TNC, CD34? Do you look at more common HLA units so you're not depleting kind of the more rare ones?
So basically, the main parameter we use is TNC content. You have to remember, MD Anderson is in Texas. So people in Texas are big. So we look for the core units that have the highest cell content in order to be able to start with a product that has sufficiently high NK cells. That is our main selection criteria. We have an algorithm. HLA does not play any role in the selection of the core blood unit.
And that threshold for you guys, can you talk a little bit about that? Is it above, 1.1, 1.2 billion? What's kind of the sweet spot for you for those units? Or what is the minimum spec that it has to be in order to qualify for you guys?
So when we start with a large core blood unit with, and I don't recall the threshold number of TNC, we even have started to expand one core unit. I use it in 2 patients. That was particularly in the times of the pandemic, when we were a bit up the creek. Now the problem is that we don't end up with, in many cases, with enough NK cells, and the dose of NK cells that we want to infuse are the dose defined in the clinical trial. So right now we try to infuse 1 times 10 to the 8 per kilo, and using what we call BOGOs. Buy one, get one. We don't get enough NK cells in many cases. So that's, now that the pandemic is over, we're back to using 1 core unit per patient.
Okay, Thank you.
Okay, I think we're literally right on time. I want to thank you for that excellent talk, and I want to thank everyone here for sticking around. Please stick around. I think the next session is here, the oral abstract, so please come hear the abstract presentations. Thanks, everyone.